Hello friends, welcome back to my YouTube channel Astin Padasare. So in this video, I'm going to provide you a more scoring topic with regarding to the DBT BITP examination. So if you're prepared well means just keep those topics as a checklist. If you left any topic means please do prepare those topics. If you didn't even start your preparation means just have this particular thing. Just prepare notes only for the topic that I'm going to discuss in this particular video. Okay, so all the topic are very much important. Definitely you can expect at least 50 questions from these topics that I'm going to provide you. Okay, so please believe me, definitely last time also regarding to DBT BITP 2023 also I made this particular topic list and nearly I think 30 questions has came from the topic that I had discussed. So you can follow those topics. Okay, so first is cell cycle. Regarding to cell biology, you need to follow cell cycle and importantly their regulation regarding to CDK and their respective cyclins and in which stage they are active. And next, enzyme inhibition. Both you need to look for graphs and also the competitive inhibition means what are the examples of competitive inhibition like that. Those things need to be studied. Next is Ramachandran plot. You need to know about proline, glycine, which is coming under disallowed region, which will be coming under allowed region, and also pi angle, psi angle, and all those things with regard to Ramachandran plot. Next is amino acid. So you need to know about type of amino acid, types of amino acid, and sometimes question will be asking which amino acid will be having two or more than two number of chiral center. Which amino acid has no chiral center. Like those questions also you can expect. And also biosynthesis of amino acid. So the precursor of those amino acids, from which precursor the amino acids are coming. So biosynthesis of amino acids is also an important thing. And also inborn errors of amino acid metabolism like phenylketonuria, which enzyme is lacking. Okay. Next is electron transport chain. You need to study about four complexes and the inhibitors of electron transport chain. Next is antibiotic. Okay, so antibiotics is very important. So kindly do study about those things. Next is translational inhibitor. What are the translation inhibitor? Whether it is inhibiting prokaryotic translation or eukaryotic translation, like that you need to know. So while studying translation itself, you need to cover about genetic code, universal genetic code, and some microorganism and some other organelles have different type of genetic code. So you need to study about genetic code, which is start codon, which is stop codon, and also study about transfer RNA. Okay, just study these two things in translation. No need to study about mechanism of translation. Because when you compare translation, transcription, replication is somewhat more important when it comes to DBT, VITP examination. Next is type of RNA, mRNA, rRNA, and all those RNA and their respective functions, pvRNA, like that. Types of DNA. Last time one question was asked from types of DNA. Like they are asked about the rotation. A form is right-handed, B form is right-handed, Z form is left-handed rotation. So that question was asked. Next is DNA polymerase. So you need to study about DNA polymerases, all the types and which some are involved in lagging strand replication, some are involved in leading strand replication. So you need to study about. And also you need to study about template dependent DNA polymerases and template independent DNA polymerases. Okay. Next, also while studying DNA polymerase itself, you need to cover about RNA polymerase. RNA pol 1, pol 2, pol 3. Which RNA polymerase is involved in transcribing your mRNA, tRNA, ribosomal RNA, all those things. And where RNA polymerase are located. Some are located in nucleolus, some are located in some other organs. Okay. Next important thing is that you also need to study about the alpha ammonite sensitivity. Okay, so which RNA polymerase is more sensitive to alpha ammonite? Okay. Next is biochemical pathway. So with regarding to biochemistry, glycolysis, Krebs cycle, all those things. Just study about in which organelle the pathway is taking place and what about the energy yield. How many ATP are produced, how many NADPH are produced. And also you need to study about activators, inhibitors of the respective biochemical pathway. At least try to cover glycolysis, Krebs cycle. Both are very important. Next is the cell organelle. For example, there will be one question asked in previous year like in which organelle glycosylation of proteins occurs. So you need to study about the organelle and also their respective functions or uniqueness. Peroxisome, lysosome, mitochondria, last step like DBT, BITP 2023. There was one question asked like how did mitochondria evolve? So the answer is endosymbiotic theory. Like that simple question only it will be. Just read. That's enough. Next is protein sorting signal. How proteins are imported into nucleus, where the signal is located, whether it is N-terminal or C-terminal. You need to know about KDL sequences, how protein are targeted to peroxisome. Just study about the signaling sequence alone. Okay. Next is telomere. Telomere is very important. Sometimes they will be asking you to write the structure, sorry, sequence of telomere, and sometimes there will be questions related to telomerase enzyme with relation to cancer. Okay, so give more importance to telomere. Okay, next is microscopy. Your SEM, your TEM, confocal, phase contrast, dark field, bright field, stereo. Last time one question was asked regarding to the what is the principle behind stereo microscope. So cover microscopy. Next is DNA sequencing. Starting with the Sanger, Maxim, River till next generation you need to study about it. Okay. Next is NMR, X-ray, CD. These three are very important. Not only in DBT, BITP examination. Nowadays even in CSCR, DBT, BET examination, GATE examination, you can expect questions from NMR, X-ray and CD. Next is peptide sequencing. Like which will be cutting your methionine, cyanogen bromide, trypsin, elastase, all the protease enzyme and which part of that they will be cutting. So mostly it will be a direct question. Where trypsin will be cutting in N-terminal or C-terminal, like that question will be asking. Okay, chymotrypsin, carboxypeptidase, endopeptidase, exopeptidase. Okay. Next is viruses and their genome. Definitely you can expect a question. Study about the common virus like HIV. Study about HIV, how the HIV is coming into the body, and about the virus and the cell surface receptor, and about coronavirus. What is the genetic material of coronavirus? How many of you know the answer? Please do tell me in the comment section. Virus is very important. Okay. Next is period diseases. Okay, this is also very important. Next is enzyme used in RNA technology, like your RNase, DNase, exonuclease, alkaline phosphatase, polynucleotide kinase. Next, genetically modified crops. Both genetically modified crops and genetically modified animals you need to study. Okay, because recently, last year 2022, genetically modified mustard has been approved by Indian government. So you need to refer on genetically engineered mustard. Mustard was approved by Indian government. So study about which gene has been incorporated and what is the main motive of that particular genetically modified mustard. Next is restriction digestion. Okay, which is sticky and blunt and next is isochisomer, chordomer, neo-isochisomer, like that. All those things you need to study. But also star activity of restriction enzyme also you need to cover up. Next is gene transfer technique both in plant and animal. While studying gene transfer technique itself, pay enough attention to agrobacterium. Even though if you can't able to cover up the entire technique, you can just study about agrobacterium, about a virgin, tDNA, binary vector, co-integrate vector, all those things. And also in animals also you need to study about gene transfer technique. Since it is a department of biotechnology, so most commonly biotechnology related questions will be huge amount. Okay. Next is regarding to screenable marker, selectable marker, all the antibiotic and how they are detected, whether they are detected by fluorescent way or histochemical way. So better go for refer biotechnology by Satyanarayana book. If you have that book means just concentrate on the topic which you felt is very easy. Just note down the tables that are given in that particular book. Next is molecular marker. Last time one question has came in RAPD. So study about RAPD, which is codominant marker, which is dominant marker, and which is used for DNA fingerprinting. Okay. Next is DNA footprinting and DNA fingerprinting are the standard techniques. Okay. Next again, chromatography. Both ion exchange chromatography, like cation, anion, how they are eluted, or about pH charge, and gas chromatography, reverse phase chromatography. Study about all those things. Okay. Next is PCR. So also last time two questions asked from real-time PCR. So PCR also very important. Sometimes there will be questions related to problem like they will be asking. Sometimes stack polymerase like that they will be asking. Sometimes primer. They will be giving a sequence and what is a forward primer, what is a reverse primer. Those kind of questions. Those are very simple. If you didn't know, just check out the thing. You will be finding them easy. Next is histone protein, which is a linker histone. What is histone tetramer? And all those things. Next is nucleosome, chromosome packaging and DNA replication. So DNA replication is very important. So please don't skip the DNA replication and the topoisomerase, DNA, gyrase, and about chromosomal banding technique, G banding, Q banding, C banding, R banding. Next is mutation. So you need to know about base analog and transition, transversion. So while studying mutation itself, you need to study about Ames test for mutation. Okay. Next is chromosomal aberration, both numerical and structural. So last time one question was asked from numerical chromosomal aberration, like formula for nullisomy and all those things. And structural aberration is also very important. Okay. Next, symbiosis mutualism, like lichen. What is the thing with regarding to lichens and about symbiosis mutualism? These are very important. Okay. Next is Beer-Lambert's law, the formula you need to study, and also few theory. Next is photosynthesis. Again, two questions asked from photosynthesis. Okay. So cover photosystem 1 and photosystem 2 in detail. Next is C3, C4 CAM pathway. There will be that is a tabular column is available in the internet, I think. So I was studying from that only. If you study the tabular column alone means that well sufficient. Next is plant growth hormone. Mostly they will be asking either the function or the precursor. What is the precursor of auxin hormone? Which hormone is used in the process of fruit ripening? Which hormone is involved in processing of delaying senescence? So you need to read the question correctly. Next is secondary metabolite. Both you need to study about the types of secondary metabolite, the pathway, the precursor, and also you need to study about the commercially available secondary metabolite and from which plant source they are extracted. So these questions are nowadays asking in all the competitive examinations. So of course, DBT, BITP also you can expect those questions. Next is cell line. So you need to study about the cell line which are used in commercial applications. So recently for corona vaccine, Viro cell line has been employed for culturing the virus for the vaccine purposes. So identify some more cell line and their applicability. Next is blood grouping. Recently in CSIR December 2023, question has been asked in 4 mark. A, B and O. What about things? Sometimes genetic related questions are also asked. So don't skip blood group. Blood grouping is very important. Next is pedigree analysis. You can't expect the problematic, but they will be asking few disease will be given and what is the mode of inheritance, which is coming under X-linked dominant, which is coming under X-linked recessive, like that you can expect. Next is Hardy-Weinberg equilibrium. Next is CRISPR-Cas. Last time one question was asked, they asked about the full form. What is the full form of CRISPR-Cas? Next is siRNA, mRNA, the precursor and the biochemical bioprocess. Next is gene knockout technology. You need to study about how to identify recombinant, non-recombinant, that neomycin, G418, all those things we study, ganciclovir, and also study about Cre-Lox-P recombination, like in which if it is like this means some process will be taken, if it is in this position means some process, like Cre-Lox-P recombination. What is Cremose, what is Loxmose, and about the promoter. Next is BLAST. What is the full form of BLAST and what is the like there were many programs like BLAST P, BLAST, and you need to study about all those things. Next is bioinformatics. You need to study about database, which is a protein database, which is protein sequence database, which is nucleotide database. At least study about the name alone. Next is glycosidic linkage in disaccharide. So study about lactose, sucrose, maltose, cellulose, cellobiose, like that. Next is commercial production. Commercial production means how organic acids are produced. Which organism is involved in commercial production of organic acid? Like which organism are involved in citric acid? Which organism is involved in production of vitamins? So vitamins, organic acid, amino acid, antibiotics are very very important. At the same time, you need to know which organism is involved in fermenting your curd, dosa batter, cheese, and milk. Like that, you need to know. Next is immunotechnique like ELISpot, ELISA, and age technique like agarose gel electrophoresis. They will be asking, they will be giving you a gel which will be running first, whether Nick will be coming first or which will be coming like Nick, concatamol, like that. There were many things are there. So I think you remember the diagram. So just note down the diagram. Next is STS page technique. Next is nodulation. Nod gene. A, we have studied the root nodulation process. With regarding to plant biotech, I'm just telling you. Next is nitrogenase enzyme. So we have 3 enzymes. When you studied about nitrogen metabolism, nitrogenase enzyme is important. Nitrate reductase, nitrite reductase. So you need to study about the structure. What is present, where petrin group is present, where MOF, molybdenum group is present. Like that you need to know. And also pay enough attention while studying nitrogen metabolism in plants. Next is hybridoma technology, monoclonal antibody by hybridoma technology. So very, very important. Next is vector. Next is melting temperature. So sometimes they will be asking, you need to know about the formula. Sometimes application oriented questions will be asking. Next is sterilization technique in microbiology. With respect to microbiology, I'm telling you, sterilization technique will be asked. Try to know about phenol disinfection coefficient. So questions might be asked from that. Next is microbial growth. So problem like how do you find a specific growth rate, like that they will be asking. Prescott book itself only two problems are available, so just work out those two problems. And regarding to lab practical side, they'll be asking questions from microbiological media. Okay. The next is transpose. And last time one question has asked like how do genes are transposed? Whether which enzyme is involved? So many of you had selected translocase. The correct enzyme is transposase enzyme. So like that simple question and only they'll be asking. Next is antibodies. All the antibodies, IgG, IgA, where segregated component is present, which is a dimer, which is a pentamer, how they will be running in a gel, like that you need to study. Next is autoimmunity, hypersensitivity. Autoimmunity means you need to study only the tabulated column that is given in the QA book. Next is hypersensitivity. All the four hypersensitivity reactions are very, very important. Next is MSc, both MSc class 1 and class 2. Next is your complement system. Don't go in deep about the complement system. You need to study about the three pathway and which is C3 convertase, C5 convertase, like that. Next, you need to identify the difference between prokaryotes, eukaryote and archaea. There is a tabulated column that is available in the internet itself. So just do and research about that. Next is cell cycling. Again, very important. You need to study only the tabulated column alone. Don't go in detail, because if you take cell signaling, it runs in pages in Albert book and Lodish book. Just concentrate on important part. Sometimes they'll be asking you to figure out the flow chart. Okay, G protein coupled receptor pathway is very, very important. Okay, and the example of pathway. Okay, next is cancer. Cancer will come, sometimes apoptosis related questions also will be asking. So study about cancer. Okay, and apoptosis, and also you need to find the difference between apoptosis and necrosis. These are usual things. Next, genetics, what you can figure out, just identify the examples alone. What is the example of maternal inheritance? What is the example of Mendelian inheritance? What is the example of maternal effect? Those fancy words in genetics. What is penetrance? What is expressivity? What is phenocopy? Okay, so those things are very important. No need to work problems when it comes to genetics. Just identify the terms alone, important terms alone in genetics. Next is DNA repair pathway and the respective diseases. The nucleotide repair pathway, which disease will be coming if the nucleotide repair pathway is not working, like that you need to know. And recombination pathway, like Holiday junction, rec component. I think you need to study about DNA recombination in bacteria and eukaryotes. Okay. Next is post-transcriptional modification. When it comes to post-transcription, it is again an important splicing, group A intron, group B intron, mRNA capping, polyadenylation, all those things. Next is membrane transport. Come to cell biology, you need to study about the membrane transport, sodium, potassium pump, and cytoskeleton. So if you think these concepts are very huge, there is nothing. We are already wasted many time points. So please identify notes for these topics. So at least know what is given under those topics. Transposer means just read at least read Wikipedia alone. Because in transposon and all, very simple question, which enzyme is active? It is transposase. That's what they are asking. You no need to go into detail about the transposon and all. Just set only 20 minutes for each topic. You can cover them easily. Okay. So thank you friends. Thanks for watching this particular video. If you find this particular video is very helpful, please subscribe to my YouTube channel. Thank you friends. Thanks for watching this video.